All right, great, thank you. It's an honor to be here on behalf of Neuren. Syndromic forms of autism often feature synaptic dysfunction, and today I want to tell you a little bit about how we are aiming to modulate IGF-1 signaling for the treatment of these neurodevelopmental synaptopathies. But, let's just take a step back first. The human brain consists of about 100 billion neurons, and these neurons communicate with each other at specialized junctions called synapses. Each neuron has anywhere between a few to hundreds of thousands of these synaptic connections, and you can begin to appreciate that here in this picture of a neuron that's labeled with a presynaptic protein in red and a postsynaptic protein in green. So every yellow dot you see is a synapse. And the strength of these synaptic connections can be modified, and the ability of neurons to change the strength of their communication with other neurons is called synaptic plasticity. And this synaptic plasticity is the foundation of learning and memory. So if we zoom in on one of these yellow dots in a cartoon form, it would look like this, where you have one neuron releasing neurotransmitter that's detected by receptors on the postsynaptic neuron. And many neurodevelopmental disorders are caused by genes that affect this synaptic transmission and plasticity, and that's why they're called synaptopathies. There are typically no gross structural brain abnormalities, just trouble in the way that the cells are communicating with each other. So restoring synaptic activity might be a way that we can treat some of these neurodevelopmental disorders. And what I'm showing in the box are just some of the many genes that have been linked to these neurodevelopmental disorders, and we're going to start with SHANK3. So Phelan-McDermid syndrome is a neurodevelopmental disorder that is a severely debilitating lifetime condition for which there are no treatments. Most common symptoms include impairments in communication, social interaction, motor function, cognitive function, and activities of daily living. There are also a number of other symptoms that affect people, including seizures, sleeping difficulties, GI dysfunction, etc. And, to put that in a little more context, I'll just read one of the excerpts from the Voice of the Patient report. PMS has severe quality of life impacts on those living with the disease, as well as on parents and siblings. Most activities of daily life, including communicating needs or wants, self-care, which includes bathing, dressing, toileting, and socializing with peers and siblings, are affected. Most individuals living with PMS rely on their parents and caregivers for all of their daily needs, and many require 24-hour care. The good news is that we actually know a lot about the gene involved in PMS, SHANK3. So again, here's another cartoon of a synapse, and you can see SHANK3. It is a scaffolding protein, and its role is to connect neurotransmitter receptors, ion channels, and other membrane proteins to the actin cytoskeleton. In PMS, either a deletion or a mutation in the SHANK3 gene results in less SHANK3. And when there's less SHANK3, then there are fewer of those receptors to detect neurotransmitters on the postsynaptic neuron. And that leads to impairments in synaptic transmission, which we think are fundamentally the basis of the disorder. In mouse models, you also see that deficits in SHANK3 lead to PMS-like behavioral abnormalities, including repetitive grooming, social deficits, anxiety, and motor abnormalities. So again, haploinsufficiency of SHANK3 appears to underlie the manifestations of PMS by impairing synaptic transmission. So now a little bit about IGF. So IGF is insulin-like growth factor, IGF-1. It has a lot of different functions within the brain and even throughout the body. Through its receptor IGF-1R, it induces multiple signaling pathways that promote cell survival, modulate inflammation, and regulate synaptic transmission. Interestingly, IGF-1 levels are actually lower in the CSF, the blood serum, and the urine of children with autism spectrum disorder in general. And what I'm showing here on the left is a table of concentrations of CSF IGF-1 and IGF-2 in patients with autism. You can see that there's no difference in IGF-2, but there is a significant reduction in IGF-1. And that's shown on the right as well. So there is a correlation between the amount of IGF-1 in the CSF with age, and in the black dots are the autistic kids, and you can see that in general, those levels are lower than their typically developing controls. So several lines of evidence suggest that if we increase IGF-1, we may be able to reverse some of the deficits in synaptic signaling that underlie the disorder. This is some work from Joe's lab showing that in a mouse model of PMS, on the left, what you can see is that if you induce long-term potentiation or a synaptic strengthening protocol, the PMS mice, which are labeled in the open circles, show a smaller response than the controls. When you add in IGF, you reverse that deficit. And this even translates into behavioral fixes. So, as I mentioned, some kids with PMS have, motor deficits. There are subtle motor deficits in the mouse model as well, and IGF reverses it and even enhances motor performance to some degree. Second, if we take neurons derived from patients with PMS, we see clear phenotypes, again, that show that there are deficits in synaptic transmission. So on the left, you can see that there are fewer action potentials, less neuronal firing in the PMS patient neurons, and you can also see that the excitatory postsynaptic currents are smaller, the amplitude is smaller, and the frequency of those responses is smaller. Again, if you think about the number of receptors in the membrane, it makes sense that if there's less SHANK3, there are fewer receptors, there are these deficits. On the right, you can see that, again, when you treat with IGF in these cultured neurons, you can reverse that phenotype. And the third piece of data comes from Alex and Joe and team, and a couple of pilot studies that they have done with IGF, showing that IGF improved sensory reactivity symptoms, repetitive behavior, and hyperactivity in PMS children age 5 to 9. This was 2 small clinical trials, and this is the paper sort of summarizing both of those. I'm sure Alex will might go into more detail on that, right? A little bit, okay. But we'll move on from there. So a little bit about NNZ-2591. So this is Neuren's drug for neurons. So NNZ-2591, it's a small molecule. Why don't we use IGF-1? We could use IGF-1. There's some great pilot data that shows that it works. It doesn't have as good a drug-like properties. So with NNZ-2591, we have very good oral bioavailability, and there are some state-dependent changes that we can regulate IGF-1 activity with that you will see here in this schematic. So IGF-1 activity is really tightly regulated, and it's regulated by these IGF binding proteins that bind IGF-1 with really high affinity and prevent it from binding to its receptor and initiating signaling. Our drug basically prevents IGF-1 from being bound by these IGF binding proteins, and that frees the IGF-1 to bind to its receptor and increase signaling. So the hypothesis then is that by increasing IGF-1 signaling, we can restore some of the synaptic dysfunction, reverse some of the synaptic dysfunction that underlies these synaptopathies. And don't pay attention to the details here. You couldn't if you tried because it's too small. But basically, this is just showing the dose-dependent reversal of behavioral abnormalities in the PMS mouse model, as well as some reversal of morphology changes in these models. So we conducted a phase 2 trial in children with PMS. This was a small study of 18 subjects, ages 3 to 12, open label. There are a number of endpoints that Alex and other clinicians helped us to develop. Paige touched on this earlier, where we need to have very syndrome-specific measures to help us understand if there's any change for these kids. And what we found is that we did see efficacy across a number of measures. So across a clinician global impression scale, across caregiver impression, across different various measures of communication, behavior, GI health, quality of life, and sleep. And I'm going to go into detail on just a couple of these measures. So this is a waterfall plot on the left of the PMS-anchored CGI overall score. And so what I mean by that is clinicians are rating the patients after drug with a checklist of things that specifically affect these patients, so PMS-anchored. And what you can see is that 16 out of the 18 showed improvement. And on the right, you can actually see the domains that moved. And all of the domains moved. The domains that moved the most were expressive communication, cognition and learning, and receptive communication. So all things that are critically important to the patients and their caregivers. This, what was nice to see is that in addition to the clinicians rating the kids, you also see that the caregivers came up with a very similar score, noting that 15 out of the 18 improved, and again, the biggest movers were communication and cognition learning. There is a place where both the clinicians and the caregivers can note improvements that they see in the CGI or the CIC respectively. And I won't read all of these, but it's interesting to us that we see common themes emerge. One of the clinicians noted that they were able to, the participant was able to focus and expressive communication, gross motor skills, those are the things that are improving, and that what was noted in the CGI. And similarly for the caregivers, again, more focused, engaged, aware of their environment, people. So we will be starting a phase 3 trial in PMS participants ages 3 to 12 years old mid-year. Hopefully within the next couple months. This is going to be a placebo-controlled trial, 13 weeks, and afterwards there will be an opportunity for open label extension for those participants who want to participate in that. Okay, I'm going to talk about 2 other disorders where we also have some preliminary data on 2591. So we get asked the question, do you think 2591 is going to work on all of these neurodevelopmental disorders, or at least all synaptopathies? No, we definitely don't. And we have preclinical data showing that, NNZ doesn't reverse synaptic dysfunction in certain models where you modulate some genes, but it does work for others. 2 of the ones where we think it might work well are Angelman syndrome and Pitt-Hopkins. So Angelman, it's been mentioned today, is an additional neurodevelopmental disorder, again, where there are no approved treatments, but there are several ASOs and other potential treatments in clinical phase testing right now, so it's an exciting time for that community. Similar to PMS, the common symptoms include impairments in communication, challenging behaviors, sleep, motor function, and cognitive function. And, here's my schematic of another synapse. And Angelman syndrome is caused by haploinsufficiency of a protein called UBE3A. This is a ubiquitin protein ligase, and it basically targets other proteins for degradation. You can see it in the blue circle in the middle of the synapse. So one of its key targets is ARC. And ARC is a synaptic protein that promotes internalization of AMPA receptors at the surface. When you have less UBE3A, this leads to an increase in ARC expression and more AMPA receptors being pulled into the cell. And so again, here we have haploinsufficiency leading to impairments in synaptic transmission, and that's what we think is underlying the disorder. So we conducted a small phase 2 in Angelman syndrome, very similar to what we did for the phase 2 in Phelan-McDermid. And we saw similar results. Here we saw 11 out of 13 showed improvement according to the clinicians. The domains are shown on the right. Here we see communication, behavior, and gross motor were all significantly improved. And this was consistent with what was seen with the caregivers, where 8 of 12 showed improvement, and again, cognitive abilities, communication, and motor abilities were the things that moved the most. And what I should note is that in this study, unlike the Phelan-McDermid study, we did have older children. When we look at just the children ages 3 to 12, they all showed improvement. And so again, getting to the point where we do think more improvements will happen in the younger kids, but we still think improvements can happen in the older and even adults potentially as well. So this is the Bayley-4 scale across a number of different domains: cognitive raw score, expressive communication, and receptive communication on the top row, showing about 50% and 60% improvement respectively. So it was great to see that. And again, multiple clinical endpoint measures showing movement in the same sorts of domains. And again, this is when you hear from the clinicians and the caregivers, and they're saying things like much calmer, focus is better, hand flapping stopped, and the caregivers are saying more focused, balanced, concentrate longer, you're seeing these same themes come up over and over again. So I know this has been a whirlwind tour. I'm going to quickly go through Pitt-Hopkins. Again, it's some of the same themes. Very similar common symptoms, so I'll just go to the next slide. Pitt-Hopkins is caused by haploinsufficiency in a transcription factor called TCF4. TCF4 regulates the expression of a number of genes involved in synaptic transmission, including synaptic proteins and ion channels. When you have less TCF4, as you do in Pitt-Hopkins, you have deficits in synaptic transmission, network excitability, and homeostatic plasticity. So again, haploinsufficiency leading to synaptic dysfunction that we believe underlies the key aspects of the disorder. So this will look familiar now, another phase 2 trial, right, in a small cohort. Here again, we're going 3 to 17 years old instead of just the 3 to 12. And we saw, again, that we saw efficacy across several measures that are relevant for Pitt-Hopkins specifically. So an improvement from both the clinician perspective as well as the caregiver perspective, and in terms of the severity of the syndrome and in terms of the top 3 concerns that caregivers have. Here are, again, some waterfall plots. Here we're showing 9 out of 11 showed improvement, and some domains moving, but of course much more heterogeneous here, but increased improvement in social interaction, language communication, and gross motor. Caregivers, again, noting very similar findings, so 8 out of 11 showing improvement. And again, social interaction, motor, communication, those are the ones that rise to the top. So really seeing those themes come through each time is building confidence for us that this is doing what we think. And you'll see some similar themes again in the notes from the clinicians and the caregivers. Improved expressive communication, 2 additional words, uses AAC device to ask for food, increased vocalization, able to match items, more calm and attentive. So again, similar themes coming through. And so, in summary, what I hope you could appreciate from this talk is that, Many of the syndromic forms of autism are caused by mutations in genes that are critical for synaptic transmission. There are no disease-modifying therapies for the majority of these disorders, and they have significant unmet medical need. Cell-based and animal models have demonstrated that reversing synaptic dysfunction may lead to improvements in function, and that there is a therapeutic window postnatally. And there are several lines of evidence that increasing IGF-1 can correct the deficits in synaptic signaling. So by correcting that synaptic dysfunction, we believe our drug might improve the lives of some people with certain neurodevelopmental disorders. This is a safe and well-tolerated drug. It's taken orally, and we will continue to push these forward and really want to thank all of the patients who have participated in the trials. And the last point, all of this would really not be possible without the help and support of the academic medical centers, clinician partners, patient advocacy groups, and the participants in the clinical trials. If you noticed, I don't think I really called it out, but most of the data, the lines of evidence indicating that modulating IGF-1 signaling could be useful came from the Seaver Center and their basic research and then their clinical research. And when we talk about the lack of funding, it doesn't just affect academia, it's going to affect the entire ecosystem because we rely so closely on the advances in the basic findings, as well as the clinical research that's being done here to find new endpoints and to find new biomarkers that we can use to test our drugs. So a big thank you to everyone at the Seaver Center who works with us. It's been fantastic, and we have a lot more to do. So thank you.